So it is possible a change in the DNA can cause an altered form of the expression of the DNA, and this new expression may cause incomplete dominance or it may cause codominance. Now what is codominance? So let's start the new topic, and that is also explained in the NCERT. Remember, in the first class I explained some of these things to you. So now we have to pay attention to this last point. If you remember, in the case of the human, we learned various types of blood groups: A blood group, B blood group, AB blood group, and the O blood group. So this topic is dealing with the human blood groupism only. So let's understand the basic again, because I want you to be clear with this concept. So human blood group is controlled by a gene present on the 9th chromosome. So remember, we have 23 chromosomes, and the 9th chromosome has a gene which controls the blood groups. Now blood groups are nothing but the sugars. On the surface of the RBC there is an antigen. Remember, antigen antibody topic. This was something which I explained to you in the 11th class. I'm talking to the 11th O basically, because I have explained these things in detail to you. And the baseline which I told you was donor's antigen should never match with the recipient's antibody. So if you remember this concept, we learned about which one is universal donor, which one is universal recipient. So just go through it once. A can give blood to A as well as to AB. B can give blood to B as well as to AB. AB can give to AB. And O can give to A, B, AB as well as O. So universal donor is O, and the universal recipient is AB, and rest all can either give to A or give to AB, or can give to B or give to AB. So now the same concept, which I won't be discussing anymore, is supposed to be dealt in the form of genetics now in your 12th class. So the topic is the codominance. Now let's understand what is this A, B, C, D, A, B, and AB as well as O. So human RBCs in the blood carries on their surface. Now remember, animal cells carry a glycocalyx or cell coat. So the cell coat is made up of mucopolysaccharide. Now on the surface of the RBCs are present antigens or sugar. Now these sugars are otherwise called as mucopolysaccharide or polysaccharide. Now the whole sugar is similar except the terminal sugar. So it's a long chain, and in that long chain the last sugar has undergone modification, and accordingly various blood groups are there. So the terminal sugar molecule of this carbohydrate in the case of B blood group is unmodified form. It is alpha D galactose. Galactose sugar is a 6 carbon sugar. And remember, D means dextrorotatory, 11th class topic, so turning the polarized light towards the right side. And alpha means when you make a pyranose form or furanose form of sugar, if you remember that concept, O is present here. If the OH is present on the opposite side, it is called as alpha. On the same side, it is called as beta. So in the case of the blood group B, the sugar is unmodified one in that mucopolysaccharide, and it is alpha D galactose. But in the case of the human blood group A, it's a modified form, which is the terminal sugar is N-acetyl alpha galactose amine. Galactose amine means there will be a NH2COCH3, NHCOCH3. This is the way you represent the amine. Amine group is mentioned in this manner, galactose amine. So this is called acetyl, this is called the amine, and then galactose will be shown here. So please remember, A type and B type, two sugars are there, and now books will simply call them as sugar type A, sugar type B. Now what exactly happened is, in the case of a blood group O, there is no sugar present, so no antigen present or no mucopolysaccharide present on the surface. That's why the blood group of the O person is considered as O, because antigens are absent. And in the case of a person who has blood group AB, both sugars are present. So the topic of codominance is dealing with the blood group AB. So what exactly is the basic behind it? The genetics is like this. There is a gene on the 9th chromosome called as the antigen gene or the I gene. I gene is also called isoagglutinogen or agglutinin gene, also called as agglutinin genes. So these genes are of three types. Remember, human blood is an example of, again, something, see, we are learning in this topic is that how Mendel was wrong to some extent. So Mendel said dominance. We learned a few minutes back incomplete dominance. Now we are learning codominance. One more thing Mendel said was that each factor has two forms, and that means two alleles. But what you are learning now in this topic is there are some genes or factors which have more than two alleles. Such a mechanism or process is called as multi